Hello everyone. So I hope we are audible to each and every participants joined to us on YouTube live. Please write in the chat box that you are audible. We are audible or not. Okay, everyone. So we'll start in a minute at 4:1, exactly at 4:1. So thanks for your patience, and we'll start soon in a minute. Thank you. Okay, so we can start. The host, you can proceed ahead. Thank you so much.
Hello and good evening to our speaker and our participants for the evening. A warm welcome to everyone present here. To begin with, I'd first like Dhairya, the founder of Scientific Community, to give a brief regarding Scientific Community. Over to you, Dhairya.
Yeah. Thank you so much, Shruti, for giving this opportunity to actually introduce the scientific community to each and every participants today over here. So I would start with the community story, that how it started in short. That like our community actually started on WhatsApp, and it's still prevailing upon the WhatsApp groups. And the link for joining our community would be given to you all during the session only. So firstly that, and what our WhatsApp groups actually serves is that it creates a pool of informations for each and every participant, for making it as an interdisciplinary open platform for each and every one. Let it be research scholar, professor, or a undergraduate student. It's open platform for everyone, first thing. And now I would like to introduce the features of the scientific community. Right. So scientific community has a wonderful feature for which we even have created our website, and that feature is that you can give your blogs or articles or science art. If you really make beautiful science art, then you can actually send us your science arts, your science articles. Let it be opinionated articles or uh, uh, editorial articles or opinionated blog. We even publish editorial blogs. So you can send it on our email, and you can find that email in our chat box soon. So you can send your articles, your science art, your science memes also to get published on our website. You can visit it easily. So it's completely free of cost. There is no processing fee. You can even send your rejected review articles to us. We have an editorial team and advisor team which actually helps you to actually increase your efficiency more in scientific writing and also science art. So we can even guide you by connecting to the experts. So that is one thing. The second thing is PhD connections. So we have lots and lots of openings all over the world going on right now during the pandemic period and also after the post pandemic. So we actually offer the open positions that are there, that are prevailing in the universities and research centers all over the world. So you can connect us on the Twitter and also join our community groups. On every Monday and Friday, we actually tweet all the open positions. Let it be junior research fellow position, senior research fellow, postdoc positions, or a PhD position. And we even give you open job openings also. So that goes for PhD connections. And after that, we have a feature of newsletter where you get all the beautiful insights of research articles and other informations which actually uh, prevails in the science field, and encircling each and every fields. It's just not restricted that it's just life science or restricted to one field like biotech. It even have maths, chemistry, physics, so you can just read that short summary and you can get insight to latest researches going on. So you can subscribe your email. It's also for free. You can just subscribe it in our website, so you'll get a newsletter every month in your inbox, Gmail inbox. After that, moving ahead, we have a feature of side chat, which is really beautiful, and I personally as a founder like it, because the main motto to actually make this platform was to create an open chatting or open networking platform, which is actually served by side chat. So we have a side chat discussion forum that also you will get the link on our website, so you can go and visit it. And you can join our side chat groups, and we even also have our side chat community, and people really discuss openly the science, and even they discuss many things apart from science. So that is the main feature of side chat. And we even conducted one or two virtual uh, discussion. And after post covid era, I think we are going to uh, perform offline activities also. So I think that would be really helpful, offline discussion clubs or something like that on science and stuffs, right? So I think please do join our community. And also today's speaker, he's also really closely associated, Dr. Gopal sir. So he's also associated with us by many ways in our community. So I would like to thank Gopal sir for really appreciating us and joining today as a guest speaker, and accepting our invitation of scientific community to actually have this webinar. So I hope that let's now actually participants are also waiting. So like, let's start, and do join our community, everyone. Yeah, sure, we would really happy to share you the link for WhatsApp group. We'll share the link for the form at the last. You can just fill it up and you can join our community. You'll get the email of joining link, right? So that's enough for the community, and I think Shruti, you can take over. Thank you so much.
Thank you, Dharya. I'm sure that we have enthusiastic participants who will definitely contribute in making scientific community a better platform. Okay, so in recent years, there is a lot of stress on sustainable development in all sectors. One of the example is, bioluminescent trees, or you can say insulin injections. So how do you think this has been possible? If you think that all of this has been a gift of recombinant DNA technology, you're right. And we are very fortunate to have with us Dr. Gopalji Gopal, who has an experience of about 14 years in the field of recombinant DNA technology. Sir completed his MSc in biotechnology in 2005 from Gujarat University, Ahmedabad, sponsored by DBT. After successfully completing his masters, he joined as a PhD at JNU in the field of molecular medicine. He was later awarded the DS Kothari Fellowship of UGC for characterization of putative type 4 secretion system of Bacillus anthracis. In 2013, he was appointed as an assistant professor in the Department of Biochemistry at Maharaja Sayajirao University of Baroda, and was at the department for 2 years. Currently, Sir is an assistant professor at C.G. Bhakta Institute of Biotech, UCA Tarsadia University, and a full-time principal investigator. Sir, I welcome you on behalf of scientific community, and you can begin with the talk. Over to you, sir.
Thank you. So close your presentation. Thank you, Shruti. And good evening, everybody present here online. And I'm thankful to scientific community and its entire team for taking this initiative, especially founder Dharya Rajguru. And I'm happy and I'm proud that Dharya Rajguru is also my student. I get opportunity to teach him for few classes. So with this, I'm starting my talk.
Sure, sir. Sure. Thank you so much, sir.
Just wait. Slide is visible. Hello. My slide is visible.
Yes, sir. It is visible.
Okay. Okay. So my topic is strategy for the production of recombinant protein in E. coli. So why we need protein? First question is, before going to discuss on recombinant protein, first we should understand why we need protein. So proteins are used as therapeutics. You all are aware of about this growth hormone, insulin, vaccines, therapeutic other proteins. Many proteins are used as enzymes for industrial purpose, for research purpose like restriction enzyme, polymerase, protease, lipase. There are several proteins we use. For research also, we need good amount of protein with high purity to characterize that protein. So as you know, nowadays genome sequencing become easier. There are instrument sequencer are available, next generation sequencing platforms are available. So we have millions and billions, or you can say trillions megabases of sequence known, but we don't know the protein coded by that gene and their characteristic. So for that also, to characterize the protein, we must have recombinant protein, good quality of protein to characterize it. So what are the source? How can we get that protein? Whether this is industrial protein or therapeutic protein. So there are two main sources. Either we purify that from endogenous sources, like insulin, we can isolate from pancreas, or some plant protein from plant or from bacteria, we can isolate. So we can isolate from endogenous sources, but there are serious limitations in endogenous sources. Quantity, suppose if we isolate the insulin from pancreas, how many pancreas we have to isolate? How many cattle we have to kill, or we have to take, you can say, dead human or dead animal. So quantity is the problem in endogenous sources. Purity also, because if it is in a small amount, other thousands of proteins are associated with that your desired protein. And third is consistency. So suppose if we wish to isolate some protein, mango protein from Gujarat, and if someone wish to isolate that same protein from plant of the Bihar, mango plant from the Bihar, then that protocol will vary. You may or may not get same amount of protein with same protocol. So there is problem in consistency. So these are serious problem in endogenous sources. So second option is applying recombinant DNA technology, molecular cloning. So this is considered as a better source for isolation of protein. As you know, these all are recombinant proteins. Nowadays, hundreds of therapeutic recombinant proteins are available in market. Companies, several pharmaceutical companies have produced therapeutic proteins, recombinant therapeutic proteins. And this list is up to 1999. We have taken this list from T.A. Brown. As you all know, how we clone the gene and how we produce the recombinant protein, but this is, I'm showing you overview of cloning. So we isolate the plasmid, or you can say vector, from the E. coli, and we isolate our desired gene. So there are different process for isolation of, we will not discuss, but we must isolate that desired gene, and then we ligate both. After digestion with compatible restriction enzyme, we ligate our desired gene with the vector, and then this becomes recombinant vector. Then again we introduce this recombinant vector into the host E. coli, where that vector will amplify, because that vector, that plasmid have all the characteristic of autonomous replication. So that vector will amplify. So your gene inserted, your DNA inserted in that vector, it will also amplify. So there are two basic purpose of cloning. Either you need more amount of that same DNA fragment, same DNA you need in more copies, Our second purpose is to produce a recombinant protein, means protein coded by that gene. If you wish to produce the protein coded by that gene, so there are second purpose. So we harvest the protein coded by that gene. So there are two, you can categorize purpose of cloning into two parts. One is only we amplify the gene, or second is we try to get the protein coded by that gene, that DNA. So these are main players of gene cloning. Passenger gene, as you know, without this gene, this is main player, passenger gene, desired gene, or you can say gene of interest. Vehicle DNA means vector. So we can also divide vector into two main categories, cloning vector and expression vector. So we will discuss on expression vector, not on cloning vector. All expression vectors are cloning vector, but not all cloning vectors are expression vectors. So we will discuss about this in brief. An enzyme for ligation, for digestion, for manipulation of DNA, we need that enzyme. And then host organism, in which we add that recombinant vector, we introduce our recombinant vector, and we try to produce a copy of that vector, or we try to express the gene present in that vector. So these are some hosts. So we will discuss only on bacterial host, E. coli host. These are main players for gene cloning. Now, why we prefer E. coli for production of recombinant protein? E. coli host is preferred. We have discussed several. You can use any host for production of recombinant protein, but generally we prefer E. coli. Why? Because easy to culture. We can culture it easily in laboratories. No special equipment is required. You can use in common way that flask. Very short life, as you know, 20 to 30 minutes is generation time for E. coli. So you can amplify it, it means you can duplicate it, and you can get more number of that bacteria. And its genetics is also known. Several genome sequences known, several genes known. So it's easy to manipulate as per our design, as per our requirement. And cloning is technically easier. Transformation, selection, all these things are technically easier. That's why scientists prefer E. coli, because its culture media is also cheaper. So production cost will be less. If we produce recombinant protein in E. coli, so production cost is very cheap. But we have all these beneficial things, but we have also limitations. If we use E. coli, normal E. coli as a host for production of recombinant protein, so we have serious limitations also. Sometimes we don't get desired amount of expression of cloned gene, or we get very less amount of expression or no expression at all. There is no post-translational modification occurs in E. coli. As you know, glycosylation, phosphorylation, these post-translational modifications are characteristics of eukaryote, and bacteria is prokaryote. So if our protein required glycosylated protein, glycosylation for its activity, then that protein we cannot produce in E. coli. Disulfide bond, this is also a post-translational modification. So in E. coli cytoplasm, we cannot produce disulfide bonded recombinant protein. So if our protein requires disulfide bond formation for its proper functioning, proper folding, we cannot use E. coli cytoplasm. And last is very serious problem, inclusion body formation. So protein forms an aggregate, insoluble aggregate particle like particulate in nature on our expression. So this is serious problem. So we have to isolate our protein from inclusion body. So you may or may not get its properly folded form. So inclusion body formation is serious problem in E. coli. So we should avoid. So how scientists have during this after initial invention of recombinant DNA technology, how scientists have solved all these problems gradually. So we will see. So before discussing the strategy as per our topic of talk, you should understand how we express, what are the steps of producing recombinant protein, how we express, how we induce our gene for expression inside E. coli. So we will not discuss cloning, but after cloning, we get transformed colony. So we ligated product, we transfer it into cloning host, E. coli cloning host, generally DH5 alpha and DH10 beta we use. So after transformation, we get this type of colony, transformed colony. From this colony, we screen the positive clone. The cell, the colony which harbors our positive clone. You must have read about this replica plating, blue-white screening. So we ensure the proper orientation of our gene in the vector, and we must ensure whether our gene has been inserted or there is self-ligation of the vector. So we ensure positive clone from this transformed colony. So nowadays we use directional cloning. So we don't use that replica plating or blue-white screening. We generally use colony PCR, using that primer sets against our gene which has been cloned in the vector. And we can also confirm that or we can authenticate that positive clone using double digestion method. So now we how we perform expression. So we isolate recombinant plasmid from the colony harboring positive clone. So from positive colonies, we isolate the plasmid. That plasmid must be positive plasmid, positive clone in which our gene is in proper orientation, properly cloned. Then we transform E. coli expression strain with this plasmid, which we have isolated from cloning strain of E. coli. Then from that transformed colony, next day we take 1 to 4 colony from transformed plate and inoculate that colony in 3 to 5 ml LB plus corresponding antibiotic containing media and incubate again for overnight. Then we take overnight grown culture and inoculate in fresh media again. After reaching the 0.6 to 0.8 OD in second day, we add this IPTG if our gene is cloned under that lac promoter, you can say, or lac under lac means IPTG act as inducer for lac promoter. It's analogous to lactose, but it will not be metabolized by lac operon. So we use IPTG as inducer. So we add IPTG, and before adding IPTG, we separate uninduced culture 1 to 2 ml from that main culture, and we again allow it to grow for 3 to 4 hours or sometimes overnight. And then after this induction period, we take some sample from uninduced culture and induced, in which we have added IPTG. We call it induced culture. So we take it and then directly we lyse this cell, adding that SDS buffer, and then we load on SDS page. So minus indicates negative IPTG, means uninduced culture, and this is positive. Since we have taken 1 to 4 colonies, we check all colonies. So we have here used 2 colonies in which this is uninduced, this is induced, this is uninduced, this is induced. So through this SDS page followed by Kumasi staining, we check whether we get induction or not, whether we get expression or not. Because you don't think that if you have cloned your gene successfully, you will get expression of your gene and you will get your protein, recombinant protein. You may or may not get. So this is overall step of expression, and to check the induction expression, or you can say, since we have added this inducer IPTG, we can check this induced culture. So you can see this band, this thick band, that means this culture is also of E. coli, this culture is of also E. coli, but in this culture we have not added IPTG. Here we have added IPTG. So you can see that all bands are same except this band, and this band is corresponding to the molecular weight of our gene, coded, means our protein coded by that gene. So in this way we can ensure whether we get induction or not. Fine. So this is overall expression step. Now for expression we use BL21 basic strain. This is basic expression strain. For cloning we use DH5 alpha, DH10 beta. we cannot directly express our gene in that DH5 alpha or DH10 beta, because that strain doesn't have that characteristic of expression. We frequently use this BL21. This is basic E. coli expression strain. So what is the characteristic of this strain? So this is B strain having this 2 protease gene deleted, mutated, so that this strain is called protease deficient strain. Means why this protease deficient strain is required for expression? So that your recombinant protein remain stable inside E. coli. Otherwise, if you will use normal E. coli strain, your protein, recombinant protein, will be degraded by presence of this protease enzyme. That's why scientists have created this strain. This is genetically engineered strain in which they have mutated this 2 protease coding gene. So this is basic strain for expression. Now we have discussed the common steps of expression. Now we will discuss the strategy to get good quality of recombinant protein with good amount. Sometimes we get good quality, but we don't get sufficient amount, then cost will increase. That may or may not be viable process. So we will discuss the problem and then how scientists have solved this. So whenever you will face the problem of non-expression of your gene, then you can apply based on your requirement, based on your problem. So we will discuss how can we solve the problem by changing the host strain, by changing the vector, by changing the culture parameters, and by changing the by using co-expression, and by altering the gene sequence without altering the protein sequence. So we will discuss all these strategies in brief, so that in future you can apply this strategy whenever you will face this problem of non-expression. Because most of the time when we clone, we may or may not get expression of that gene inside E. coli, so we will not get that recombinant protein. So all our PhD will halt, because our PhD, if our PhD is dependent on that recombinant protein, and if we are facing problem in production of recombinant protein, then how can we proceed further? We don't have that recombinant protein, then forget about its characterization and its use as a therapeutic or as a vaccine. Fine. So now we will discuss one by one. So as you know, we use PET28. Just for example, I'm starting from this frequently used vector. This is also like E. coli. PET vector is choice for a researcher. We prefer this vector for expression of recombinant gene. This is PET28 vector. So this is common choice because it has a small tag, and purification is also robust. So this vector, if you will clone your gene in this vector, your protein will be conjugated with polyhistidine tag. This tag will act as a purification tag, polyhistidine, and this vector has T7 promoter. So we will discuss briefly about expression vector in second strategy, but keep in mind this vector have T7 promoter. This is strong promoter, and this is affinity tag. So if we use this vector, then we cannot use BL21 normal BL21 expression strain, because that BL21 expression strain doesn't have this, means E. coli RNA polymerase cannot recognize this T7 promoter present in this vector under which your desired gene is cloned. So that BL21 expression strain cannot be used if you have used this vector, if you have used this vector for expression of your gene. So scientists have created second strain that is called BL21DE3 strain. Means in BL21 they have added D3 gene. D3 gene means that D3 segments is bacteriophage T7 RNA polymerase coding gene. So that BL21D3 strain means this strain have T7 RNA polymerase gene. Now Since that E. coli strain have T7 RNA polymerase gene, that RNA polymerase gene will recognize the T7 promoter present in the vector. So you can get expression. So what is the characteristic of this strain? Since we have derived this BL21D3 strain from BL21, so lone and OMT protease are absent. In addition to that, D3 segments are incorporated in the genome of that BL21. This is called BL21D3 strain. So we should use BL21D3 strain for expression of our gene if we have cloned our gene in PET28 vector. We cannot use BL21, that is basic strain for expression. So this is mechanism of expression. You can see how, so this is T7 promoter is part of the vector, your PET vector, and this is your target gene, desired gene. And this T7 RNA polymerase gene is present in the chromosomal, or you can say genome of that bacteria. So it will produce this T7 RNA polymerase, and this T7 RNA polymerase will bind at T7 promoter present in this vector. So this gene, target gene, will transcribe, and once it will get mRNA of that gene, that mRNA will participate in translation, so you will get recombinant protein. So you will get sufficient. But what is the limitations of this strain? Initially people have used this strain, and nowadays also they use this strain have limitations. Expression is leaky. If you are using BL21D3 strain, expression of your gene will be leaky. Leaky expression means expression of cloned gene without addition of inducer. As I have already discussed that we use IPTG as inducer. So in vector we use a strong promoter and inducible promoter. So without induction also there is little amount of expression, that is called leaky expression. So this is leaky expression is not desired when your protein is toxic to the E. coli. Some proteins are not toxic to E. coli, but some proteins are toxic to E. coli. So we should avoid leaky expression. In that case, we cannot use this BL21D3 strain. If your gene produces a toxic protein for E. coli, then you should not use this strain, because due to presence of that toxic protein, E. coli cell will not grow. So if E. coli cell will not grow, then you will not get sufficient number of cell in that culture volume, that particular culture volume. So overall yield will be less. You will not get sufficient amount of protein because cell will not grow. So we should avoid leaky expression. So if your gene is toxic, you should not use BL21D3 strain. So how scientists have solved this problem? So new strain they created. In this is same BL21D3. In this background, they have added extra plasmid which codes lysozyme. T7 lysozyme we called it. So this lysozyme is inhibitory to T7 RNA polymerase. Means here you can see here. So this is normal BL21D3. So what happens if when without adding IPTG, there is little amount of T7 RNA polymerase present in this cell, which bind here and which starts transcription and followed by translation. Few molecules of this T7 RNA polymerase is present in this cell without or before induction, before adding IPTG. So there is leaky expression. So scientists have added extra plasmid in this BL21D3 strain, and that plasmid encodes lysozyme, T7 lysozyme, which is inhibitory to T7 RNA polymerase. So here T7 RNA polymerase, if whatever molecule 4, 5 or 10 molecule present in this cell, that T7 lysozyme will inhibit it. So it is not free to bind here, so there is no leaky expression. So they created this strain called BL21D3 P-lyse, in which lysozyme coding plasmid has been added. This strain will not show leaky expression. Fine. So whenever we produce recombinant protein which is toxic to E. coli, we should not use BL21D3 strain. We should use BL21D3 P lyse strain. Lyse E and lyse S, there are two variants are also available. Now, so this strain doesn't have leaky expression. Fine. Now, how to overcome effect of codon biasness? So sometimes expression of foreign gene encounters with problem of codon bias in expression host. So suppose if we are cloning our gene, which is human gene, and trying to produce recombinant protein in E. coli, then you may face the problem of codon biasness. Due to this bias, sometimes it's difficult to get sufficient amount of protein, or you may get truncated protein. Why? So before understanding the region, you should understand codon biasness. What is codon biasness? So as you can see, this is arginine coded by 6 types of codon. This all is called synonymous codon. So as you can see, AGA codon, in ideal condition, what should happen? All these 6 codons should be present in equal proportion, equal frequency in the genome of all the organism. But when scientists have analyzed, they've observed that AGA codon is present in 22% of cases, Drosophila 10%, and in E. coli it is present in 1%. While CGC is present in 39% of cases. So CGC is preferred codon and AGA is not preferred codon by E. coli. While AGA is preferred by human, but CGU is not preferred by this human. Means different organisms have different preference of sets of synonymous codon. All these codon are coding arginine only. This serine, all these 6 codon coding for serine only. But different organisms have different preferences of this codon, synonymous codon. This preference is called codon biasness. So what happened due to codon biasness? Due to this biasness, if you are using the gene of human having this AGA in more number, then when it will transcribe in the E. coli, and then it will participate in translation. So since E. coli doesn't prefer this AGA, so as you know, tRNA decode the codon during translation, so their tRNA, corresponding tRNA concentration is also very less because they don't prefer. So their tRNA concentration, if you don't prefer some food, your home have very less ingredient of that food. So that tRNA, corresponding tRNA is very less in this E. coli. But you are forcing that E. coli to produce a recombinant protein of your desired gene, human gene. So this codon is called rare codon for E. coli. AGA is rare codon for E. coli. AGG is rare codon. And that corresponding tRNA which decode this codon is called rare tRNA. So that concentration of that rare tRNA is very less in E. coli. So during translation of your desired gene, that E. coli will face scarcity of that tRNA. So if they will face scarcity, then what will happen? Rate of translation will reduce. Or if there is more scarcity, there is consumption of all the tRNA, then your protein will be truncated because it will not translate complete mRNA. So due to codon biasness, even you have everything right, you may or may not get protein, your recombinant protein, due to this codon biasness. So scientists observed that, they observe all these things, what is codon biasness, how it affects the expression, and then how can you solve this? So scientists have added rare tRNA gene in the E. coli, means extra copy of rare tRNA in that E. coli, and that E. coli, genetically engineered E. coli, is called codon plus strain. So scientists observed that few codons are rare codon in E. coli. So they produce this codon plus strain. So they produce two types of codon plus. In one BL21 codon plus real strain, cells contains extra copies of the arginine U, isoleucine Y, and leucine W tRNA gene, which recognize the AGA, AGG, AUA, and CUA codons. So it will overcome the problem of AT-reach gene. So scientists observed that in AT-reach gene, these codons are present frequently, and these codons are rare in E. coli. So if you are using the gene AT-reach gene for expression in E. coli, you should use BL21 codon plus real strain. If same way, they have produced different codon plus strain called RIP strain, RP strain, in which they have added extra copy of gene of tRNA, which is called RGU, which recognize the AGA and AGG arginine codons, and the tRNA gene proline L, which recognize the proline codon CCC. This strains overcome the bias of GC-reach gene. Means if you are expressing the GC-reach gene from human or some other organism in E. coli, then you should use BL21 codon plus RP strain. And if you are using AT-reach gene for expression in E. coli, then you should use this BL21 codon plus real strain. Means you have to shift your expression strain depending upon whether your gene is AT-reach or GC-reach. So sometimes scientists working on different types of gene, then they have to check whether this gene is AT-reach or GC-reach. Accordingly, they have to use this different strain. And these two strains are supplied by a Strategene company. So now, second company, they come with second strain, that is called Rosetta strain. So this Novagene created this, means that Novagene supply this Rosetta strain. So in this strain, all the tRNA which decode the rare codons has been added in same strain. This is called Rosetta strain, and that added through this plasmid, rare plasmid, they called it rare plasmid. So all the rare tRNA gene has been added through this plasmid in this E. coli, BL21D3 strain. So as a researcher, you don't have to shift your strain, whether your gene is AT-reach or GC-reach. You can use this strain blindly to overcome the problem of codon biasness. So nowadays, people use this Rosetta strain if they face the problem of codon biasness during expression. BL21C43 strain. So even in E. coli, we cannot produce globular protein and membrane protein. That proteins are considered as a toxic protein for normal BL21 strain, BL21D3 strain. But these scientists, when they were experimenting, when they were tried to produce recombinant protein, so they got few transformed colonies. Because of toxicity by expression of globular protein, that strain may not grow. Means you may not get transformed colony in the plate, so colony will die. But during this experiment, they found few colonies. This group of scientists observed few colony in the plate, and that colony considered as a self-mutant strain because they resist the toxic effect by globular protein production. That strain is called self-mutant strain, and they named it BL21C43 strain. Means this strain is nowadays supplied by Leucigene company, but this strain is self-mutant strain. Means experiment karte karte they observe few colonies grew in the plate, and then they analyze that why it is growing. So that colony is resistant to toxic effect of that protein. So they removed their plasmid, and nowadays they sell that cell so that anyone can purchase and use that strain for production of toxic protein. Means globular protein and membrane protein can be produced in this C43 strain. due to this mutation at this promoter region, they become resistant to toxic effect of the protein. After this, they sequence that genome of that BL21C43 strain, and they observe that there is mutation at this -10 region and this region. Due to this mutation, uh, E. coli become resistant to toxic effect of the protein. So based on that knowledge, NAV company created a second strain, genetically modified E. coli strain, recombinant strain, in which they have added titrable Ramenose promoter. Means T7 polymerase gene was incorporated under the control of this Ramenose promoter. In BL21D3, you must have seen here, this T7 RNA polymerase gene is incorporated under lac promoter. So here they have incorporated this T7 polymerase gene under Ramenose promoter. What is the benefit? So as you know, when we switch our tube light or bulb, so either you can switch off or switch on. We cannot make it dim light. Zyada ya kam nahi kar sakte. But so that is same in lac promoter. So if we add IPTG, it will express fully or it will not express. But under this Ramenose promoter, it is titrable. So you can gradually control the level of expression if your gene is cloned under Ramenose promoter. So you can control the amount of T7 polymerase protein inside E. coli. And since you can control this T7 RNA polymerase, you can control the expression of your desired gene, which is under T7 promoter in the pet vector. Fine. Arctic Express. So scientists observed that they tried to reduce the culture temperature of E. coli, in which they used to produce recombinant protein. So purpose of reducing, generally we use 37 degrees centigrade for growing the E. coli. So sometimes when we face the problem of overexpression and inclusion body formation, so that inclusion body is not desired because this is misfolded protein. So to reduce the inclusion body formation, generally scientists reduce the culture temperature. They grow the E. coli at less temperature, 20 degree, 18 degree, after addition of IPTG, after induction. So that we consider that when we reduce the culture temperature, so expression rate will reduce. So protein, whatever recombinant protein will be synthesized in the E. coli, that protein will be folded properly by the presence of chaperone protein present in the E. coli. The normal chaperone protein will take care of folding of your recombinant protein. Since at high temperature, optimum temperature, expression of our recombinant protein is more, so existing chaperone protein will not take care of its proper folding. So generally we reduce the culture temperature. But scientists observed that when we reduce the culture temperature, their chaperone activity also reduces gradually, because that chaperone acting optimally at optimum temperature, that is 37 degrees centigrade. So their activity reduces as we reduce the culture temperature. So to solve this problem, scientists have added psychrophilic, or you can say cold adapted chaperone coding gene in the E. coli expression strain. And that strain is called Arctic Express strain. Means, what is the characteristic of this strain? So when you reduce the culture temperature, since this chaperone is cold adapted, it will remain active at low temperature also, while E. coli, their own this chaperone protein will not be active at low temperature. So you should use this Arctic Express strain if you are growing your culture your E. coli expression strain at low temperature to get the soluble recombinant protein. BL21 stop. So scientists observed that in presence of this RNase gene, RNE131 gene, which codes RNase. As you know, if there is sufficient RNase in the cell, so your mRNA will not remain stable, because this RNase gene will cleave or digest your mRNA. If that mRNA will not remain inside the host for desirable time, for sufficient time, then that mRNA will not participate in translation, so you will not get sufficient protein. So scientists observed that after mutating this mRNA coding gene, stability of the mRNA increases, so that you will get more amount of the recombinant protein, and that strain is called BL21 star strain. This strain is supplied by in vitro gene. Expression of protein which require disulfide bond. So as you know, E. coli cytoplasm have reducing environment, so it will not allow formation of disulfide bond. So E. coli cytoplasm will not allow disulfide bond formation. Or suppose if you need the protein, recombinant protein, which require disulfide bond formation for its proper folding, then you have to isolate that protein and then you have to oxidize it outside the cell. You must have learned about insulin. So both peptide has been purified, and then in vitro, their disulfide bond formation, we allow that oxidation, and through that oxidation, it will form a disulfide bond formation. So this is tedious process, because E. coli will not allow disulfide bond formation due to its reducing cytoplasmic environment. But periplasm can allow disulfide bond formation. So either you transport your protein inside periplasm by adding signal sequences in your gene, signal peptide coding sequence in your gene. But as you know, periplasm have very less space. Periplasm is the space between inner membrane and the cell wall, is called periplasm. So that periplasm can allow disulfide bond formation, but that is very less. Space is very less, so you will get less amount of protein. So scientists through research come to know that due to presence of these two protein, thioredoxin reductase and glutathione reductase, E. coli cytoplasm have reducing environment. So what they did? So they mutated this two gene so that E. coli will not remain reducing, it becomes oxidizing, and that mutant strain is called origami strain. So if you wish to produce recombinant protein with disulfide bond formation in cytoplasm, you should use origami strain. In this strain, these two genes are mutated. These two protein will be absent, so its cytoplasm can allow disulfide bond formation. But after several experiments, suppose if there is only one disulfide bond formation is required in your protein, then this strain is good. But suppose some proteins require multiple disulfide bond formation, and as you know, if there is multiple possibilities of disulfide bond formation, then there are several permutations, combinations are possible. But out of several, only one sets will be appropriate. So in this strain, you may or may not get appropriately bound disulfide bond formation. So if there is only one possible disulfide bond formation is only one is possible, then you can use this origami. If multiple disulfide bond formation, suppose there are several cysteine present in that protein, then there are several permutations are possible. In that case, if you are using this origami strain, you may or may not get activity of your protein as per the amount of protein you are purifying. So In that case, if there are multiple disulfide bond formation is possible in your protein, then you should use sulfalestane, because in this sulfalestane, these two genes are mutant, means these two protein will not produce. In that background, this DSBC gene, which codes disulfide bond isomerase, has been added. This protein will ensure proper disulfide bond formation. It will only allow proper disulfide bond formation. Like saffron, it will only allow proper, which have lower energy, as you know, for proper folding. So it will screen the properly folded protein having proper disulfide bond formation. So you should use sulfalestane if your protein required multiple disulfide bond formation. If your protein have only possible disulfide bond formation, then you can use Organised. So this is Organised and this is sulfalestane. You can see these two genes are mutant. Del indicates deletion. In addition to that, this DSBC gene has been incorporated in this cell, which will ensure proper disulfide bond formation. So there are different variants of origami and sulfalestanes are available based on T7 expression, based on lyse Y. So different variation depending upon T7 RNA polymerase is present or not. In this way, different variations of sulfalestanes are nowadays available. Glycosylation. As you know, we cannot produce glycosylated protein in E. coli. So the glycosylated recombinant protein has also been produced in genetically engineered E. coli host. How? Using Campylobacter jejuni. So this group of scientists observed that in Campylobacter jejuni there is protein which have glycosylation. So they added this complete operon which codes for different protein which are responsible for glycosylation. They have added this PGL operon in E. coli to make that E. coli for production of recombinant, glycosylated recombinant protein. But that glycosylation pattern is not similar to the human protein glycosylation or eukaryotic protein glycosylation. This group, Recon et al. Group, successfully engineered an E. coli strain and got N glycosylation of the eukaryotic recombinant protein. But these strains are nowadays not available commercially. If you wish to do work on glycosylated protein production in E. coli, then you have to borrow this recombinant genetically engineered E. coli strain from this group, because whatever strain we have discussed earlier, that all strains are available commercially, means you can purchase from the company. But these two strains are not available commercially. You have to borrow. Depending upon their wish, they may or may not give you that strain. So this is overall about this expression strain, E. coli expression strain. There are more nowadays. Scientists are producing. They have not stopped making new strains, but this is overall strain we have used during my PhD and postdoc. Most of the strain I have used, but don't think that only these strains are available. I have recently also published one book chapter in which I have discussed almost 20 strain of E. coli. So this is list. You can find this list in my paper, Strategy for Recombinant Protein Production in E. coli. That is published during 2013. So you can find that detail of these strains. Now, so we have discussed 6 strategy. Out of 6, I have discussed by changing the host strain. Now I will discuss by changing the expression vector. Sometimes we may or may not have that expression strain. So how can we use that expression vector? What are the problem in different expression vectors? So now we will discuss. So you must convey me 10 minutes before the last time. Hello?
Yes, sir.
हाँ, 10 मिनट पहले मुझे बता दीजिएगा ना.
Okay, sir, no problem. So we will end it till like we have our time till 5:15, or I can say till 5:30 also we can
I
extend.
have, I have, I have several things. If
Okay.
you are ready, then I can extend up to 6 also.
Uh, okay, so I think 5:30 would be an appropriate time. So I think you have more 20 minutes.
Okay. So
Yeah.
expression vector. So in expression vector, that expression vector must have a strong promoter, inducible promoter, affinity tag, ribosome binding sites, and terminator. These 5 things must be present in expression vector in addition to the components present in cloning vector, because all expression vectors are cloning vector, but not all cloning vectors are expression vector. So based on that, we will discuss different expression vector. A strong promoter means, you can see, if your gene is cloned under a strong promoter, it will transcribe rapidly. So there is more mRNA, so there is more protein. If your gene is cloned under weak promoter, so there is less mRNA, less transcription, less mRNA, so less protein. So obviously our purpose is to produce recombinant protein. So we clone our gene under a strong promoter, and that promoter must be inducible. So when we add inducer, then only it should start transcription and followed by translation. These are some examples of strong promoter which generally scientists use in expression vector. This is, you can say, typical diagram of expression vector. This is promoter, operator, signed Dalgarno sequence, means ribosome binding sites, or you can say signed Dalgarno sequence, MCS, then terminator sequence, this selection marker, origin of replication, which is present in cloning vector also. Now, since we have discussed about this PET vector, as you know, this is common choice. This is small, it has a small tag. Only 6 histidine is present, which act as a affinity tag, 6 histidine, and it has T7 promoter. So we use frequently this vector for production of recombinant protein, PET28A, PET30, PET32. In this way, we use different series of PET vectors are available. But what are the limitations of this vector? Everything is fine, but what is the limitation? So it decreases the solubility of conjugated protein. Means if you are cloning your gene and your protein have problem of solubility, then this polyhistidine will also decrease the solubility of your protein. Then you will not get soluble protein. It forms an inclusion body. So this vector have serious limitations about solubility of your protein, because this, due to this continuous histidine, 6 histidine, it affects solubility of your protein, because this affinity tag is also conjugated with your protein, so it will decrease solubility of your protein. And if you are not getting soluble protein, that means your protein is not active. So if you're really facing problem of solubility, then you should change the vector. So you can clone your gene in female vector. So if you will clone your gene in female vector, your protein will be conjugated with maltose binding protein. This vector have maltose binding protein affinity tag, and it increases the solubility. Here it decreases the solubility. So if your protein have insoluble nature, then you should use this vector, so that this affinity tag will increase the solubility of your protein. Means it's affect the solubility of your protein, it increases the solubility of protein. But what are the limitations of this vector? It has 44 kilodalton tag, means affinity tag is very big. So if your protein, suppose if you are producing insulin protein, then insulin protein size is very less in compared to this tag. So your protein may not show its activity. This tag, affinity tag, will cover your protein. So you may or may not get activity of your protein. So you have to judge accordingly which type of vector you should use. If you wish to use PET series of vector, Then in pet vector also, you will get this pet 44, pet 43, in which your protein will be conjugated with this news tag. News means an utilization substance which increase the solubility of your conjugated protein, desired protein. But what are the limitations of this? So it has also almost 55 kilodalton. So this affinity tag, solubility tag size is very big. So you have to check which vector is appropriate for your gene. Suppose if your lab have affinity column, histidine affinity column, like nickel NTA column for purification, so then you cannot use this vector because your lab have only nickel NTA column for purification. And if you will produce this your protein in this MVP tagged, so you cannot purify your protein because you have to purchase amylose column for purification. As you know, for different affinity tag, there are different column for purification. Then if you have limitation, then you cannot use this tag. Means depending upon which type of purification column your lab have or your lab can afford, accordingly you can choose vector. So suppose if you have nickel NTA tag and nickel NTA column for polyhistidine tagged protein purification, then you have second choice, p hat vector. This p hat vector is supplied by CloneTech. So unlike the pet vector, it has non-adjacent histidine. In pet vector, all histidines are adjacent, continuous. But here, his tag is affinity tag is histidine only, but all histidines are not adjacent. So solubility will not be affected by this non-continuous, non-adjacent histidine. Means purification will be like same polyhistidine tag, but it will not decrease the solubility of your protein. You can see here. Here, this is first histidine, this is second, this is not continuous. So you can purify your protein using nickel NTA tagged nickel NTA column, and you will also not suffer with the solubility of your protein because it will not decrease the solubility of protein. This tag will not decrease solubility of your protein. PJEX. So if you will use this vector, your protein will be conjugated with this glutathione S transferase. This is also an affinity tag. So 25 kilodalton is the size of this glutathione S transferase. So this is lower, lesser than the MVP tag, and it also increases solubility of your protein. As I have told initially that major problem in E. coli is solubility of the protein. Generally, membrane proteins are not soluble in E. coli. Most of the proteins are not soluble. So solubility is the problem. In that case, if solubility is the problem, then you should choose different vector having different affinity tag, depending upon which vector you have. PQ vector. So in this PQ vector, gene is cloned under T5 promoter. As you know, T5 promoter is also a bacteriophage promoter like T7 promoter, but this promoter can be recognized by E. coli RNA polymerase. So you don't have to use BL21DE3 strain. If you have remembered, D3 strain we have used for PET expression. So here we don't have to use that D3 strain. The host of expression of gene cloned in this vector is either M15 or this vector, this host. These strains possess PREP4 plasmid. This plasmid encodes lac repressor, which ensures tight regulation of expression. Means there is no problem of leaky expression because repressor, extra copy of repressor gene has been added in this strain through the plasmid, extra plasmid. So there is no leaky expression in this strain. If you are using this PQ vector system and this strain, then there is no leaky expression, and there is no extra T7 RNA polymerase you have to add in this strain, because this promoter can be recognized by E. coli RNA polymerase. Sumo vector. So if you will use the sumo vector, its solubility of conjugated protein increases, means this tag will also affect solubility of your protein, means it will increase solubility. And second characteristic of this vector is you can precisely delete your protein without any addition of extra amino acid in your protein. So after purification of the recombinant protein, generally we remove the affinity tag, because that affinity tag may hamper the activity of your protein, or if you are using that protein for therapeutic purpose, then that tag is not allowed. Drug controller will not allow you to use that conjugated protein, that affinity tag protein, as a therapeutic protein. So you have to remove. So there is protease site in between the affinity tag and your protein. So different vectors have that protease site, so that you can delete, you can remove the affinity tag after purification. So in this vector, you can precisely remove the affinity tag. So suppose if you are using different vector, then how can you save the time? Since I don't have much time, I'm explaining different. So these are different affinity tag and solubility tag, and this is size of length of that affinity tag. GST tag 211 amino acid, MBP tag 396. This is different tag. There are more affinity tags and solubility tags are known. Third parameter. So we have discussed different host strain, E. coli host strain, different vectors. I'm not claiming that I have discussed all the E. coli strain used for expression and all the expression vector used for E. coli system, but I have discussed most of them. Same way, physical and chemical agents. So suppose you don't want to shift the strain, or if you don't want to reclone your gene in different vector, since we have already cloned and we don't have option, then scientists have tried these parameters also. Like I have already discussed, if you reduce the culture temperature, then also there is possibility of getting soluble protein. Means chances of inclusion body formation will decrease if you will grow your E. coli at low temperature. Ethanol addition. It has been also observed that if you will add 3% ethanol in that culture, you will get soluble protein. Heat shock, salt addition, benzyl alcohol addition. So different group have shown different reagents which affect the solubility of recombinant protein. By addition of these reagents, it will increase the solubility of your recombinant protein. The overall principle, what we can conclude, that these all things act as a shocker to that E. coli. So in shock, E. coli will produce more amount of chaperone. So if that E. coli will produce more amount of chaperone, then that chaperone will fold your recombinant protein properly and you will get soluble protein. That is the overall conclusion of this addition of different reagents or decreasing the temperature. After addition of these reagents, that cell, that E. coli will face this shock, chemical shock or heat shock. So it will start producing more chaperone protein. That chaperone will take care of your recombinant protein. Co-expression. So sometimes scientists try to co-express chaperone coding gene along with our desired gene, so that that chaperone will take care of proper folding of our protein, our desired protein. So desired gene co-expressed with different combination of chaperones, and they observed that they got soluble protein, because this chaperone will fold your protein properly. Because if you will try to overexpress your protein without increasing the amount of chaperone in the E. coli, then E. coli chaperone, whatever available amount of chaperone is present in that E. coli, it will not take care of your rapid expression of your protein. So your protein will not be properly folded. It will not forms its proper conformation. So it forms an inclusion body. It will go in, it will aggregate. Second option is co-expression of interacting partner. So sometimes in endogenous stage, as from this example you can see, when we produce CAG-X protein separately in separate host and CAG-T protein in separate, then both protein comes in inclusion body. But when we co-express both these genes in one host, as you can see, this is supernatant. So we observe both proteins in supernatant. So we check solubility by sonication of that culture and then centrifugation, and then we take that supernatant. So if your protein is soluble, you will get your protein in that supernatant. So we observe both of our protein in supernatant. While when we produce this protein individually in one host, both protein forms an inclusion body. So what could be the reason? So suppose one protein have some hydrophobic patches. So their interacting partner in its endogenous state will cover that hydrophobic patches. So only hydrophilic patches will remain exposed in the cytoplasm. So you will get soluble protein. If you will express it alone, its hydrophobic patches will be exposed in the aqueous environment of cytoplasm. So it forms an inclusion body. It will be aggregated. It will not be soluble. So that may be the reason, or may be other reason also. So co-expression can also be applied if you are facing problem and if you know the interacting partner of that protein. Fifth, changing the gene sequence. So if someone will say, if we will change the gene sequence, then we will get different protein. Then how it will serve our purpose? So we should change the gene sequence without changing the final product, final protein sequence. As you have seen, there are several codons which code for a particular amino acid. You know that degeneracy of codons. Several codons code for a part. So we have to change a codon in such a way that your protein sequence should not change. So one of the strategies is removal of signal peptide coding sequence. So sometimes we observe that when we remove the signal peptide and we produce the gene recombinant protein equivalent to the mature protein. As you know, what is the role of signal peptide? So signal peptide lead to or it helps in transportation of the protein. But when we produce that protein in E. coli, why we will get this transportation? So we generally remove this coding sequence and we only clone the gene corresponding to the mature protein sequence. So in that case, it has been observed that if we remove this signal peptide coding sequence, then we get more expression of that gene. Second, removal of hydrophobic patches. So if your protein have several hydrophobic patches, then you should remove that patches and clone only hydrophilic patches of protein, provided that hydrophobic patches, absence of that hydrophobic patches should not affect activity of your protein. Then only you can remove. So based on your activity, sometimes we wish to characterize only one domain of that protein, any particular protein. In that case, we can remove the remaining domain and we can clone that truncated protein only, their corresponding gene. Optimization and synthesis of gene before cloning. So sometimes we have some sufficient amount of mRNA in the cell, but we don't get corresponding amount of protein. Why? So if suppose your mRNA have Sine-Dalgarno sequence, and that Sine-Dalgarno sequence forms an hybridized with the terminal sequence of that mRNA. So it forms an loop structure. In that case, signed Dalgarno sequence is not free to bind with ribosome, a small ribosome. And as you know, a small ribosome binding is required for initiation of translation. So despite the presence of sufficient amount of mRNA in the cell, you may not get sufficient amount of protein due to this post-transcriptional, or you can say, yes, post-transcriptional regulation. Sometimes distance between a start codon and promoter sequence also decides level of translation. So there are several parameters which controls the translation, which controls the rate of translation. So sometimes there are several companies available which will optimize your gene sequence, and then they will give you optimized gene sequence before cloning. So you can only clone that optimized gene sequence. Fourth strategy, changing few rare codons using site-directed mutagenesis. Suppose what we have discussed that we should use codon plus stain if we are facing the problem of codon biasness. But if your lab cannot afford the purchase of that codon plus stain or Rosetta stain, then what are the second option? So if your lab have that site-directed mutagenesis facility, kits are also available, or through PCR also we can do normal PCR, we can do site-directed mutagenesis. So we can change the rare codon and we can add that preferable codon. So we remove that rare codon and we add that preferable codon which is preferred in E. coli. Then you will not face scarcity of that means you will not face the effect of codon biasness, so you will get sufficient amount of protein. So through site-directed mutagenesis also you can change rare codon from means you can add preferable codon from rare codon. So these are overall strategies we have discussed. As you can see, when we clone this gene successfully, but we could not get expression. So we observed that through in silico analysis, we observed that there are several hydrophobic patches in this gene. So when we observe, then we cloned this gene from this part only. We have removed this. So we cloned corresponding part, this part, which doesn't have this hydrophobic patches. So you can see here, we deleted this N240 amino acid coding sequence, 240 amino acid coding sequence we deleted, and we cloned only this sequence corresponding after 240. So after this cloning, we observed that there is expression. Here, when we cloned complete gene, we could not get expression. But when we removed this corresponding gene and cloned remaining part of this gene, here we PET 28 vector we used, and we cloned this 64 gene, PJEX64, and del indicates deletion, N means N terminal of that protein, 240 amino acid corresponding sequence we have deleted, and we cloned the remaining part of that gene. So we get expression, though expression is less, but we get expression. That means if when we remove the hydrophobic patches, we get expression. But when there is this hydrophobic patches present in the protein, then we could not get expression. So we discussed this all 5 strategies for protein expression to get good quality of protein and good amount of protein. You should try all these strategies depending upon which gene you are using, which protein you are willing to produce in E. coli, whatever facilities available in your lab. So this is overall. Thank you. Anybody, they can connect with me. I'm using Facebook, LinkedIn, I'm using Twitter. You can also send me WhatsApp message whenever you find difficulties in cloning and expression. You can connect with me, and I'm happy to help Indian research scholar and I'm trying to improve our all recombinant DNA technology field of our Indian laboratories. Thank you. If you have any question, you can ask. I request organizer to convey me all the questions. I will try to reply. Hello.
Sure, sir, sure. So thank you so much, sir, for great presentation and very informative session. And there are lots of questions popping up on from the participants. So I will ask you
Yes,
all
yes,
the questions drop wise.
yes, please.
So the first question is asked by a student that at which condition an E. coli species will produce better recombinant protein?
So in first this I showed you at 0.6 to 0.8 OD, generally we add inducer. Here, when it will go in log phase, then we add inducer, so that whatever protein we produce in E. coli, it will increase the metabolic load on E. coli. So we try to add inducer, we try to produce recombinant protein when culture growth reach up to log phase or late log phase. So we add inducer when that culture OD reach up to 0.6 to 0.8 OD. So we keep on checking the optical density that of that culture. Then we add IPTG. Hello. Are you listening?
Yes, sir. Okay, okay. So I think that's an fair answer for the question. So next question is asked is what is the main reason behind leaky expression of any gene?
Not any gene here. If that repression is not complete, that regulation, yes. See, here, if, as you know, in lac operon, that lac I gene product repressor protein is bound here. So that repression is not sufficient. That repression is not 100%. So even in repressed condition, there is little expression of this T7 RNA polymerase. Are you getting? So this repression is not complete 100% repressed. It will keep on expressing even without adding the inducer. Once we add IPTG, then its expression will rapidly transcribe and translate. But even before adding IPTG, it will transcribe, but slowly. It will cause this leaky expression. This RNA polymerase will bind here at promoter region, and your target gene will continuously express. That's why in expression vector, generally we use inducible promoter. But that promoter may not be completely repressed. Fine? Am
Yes,
I?
sir, that I think satisfactorily answers the this question also. So there is one more question that why can't membrane proteins be expressed in BL21?
Okay. BL21 or where?
Yeah, I think she
See,
wrote
see, see,
just
see, see. So membrane proteins and globular protein exact region is still not very known, but scientists are speculating that suppose if you are producing membrane protein in E. coli, so if it will produce overexpress, then it will clog its transport system. Are you getting? It has also transport system in E. coli, so that it will go inside this membrane, or it will go outside. So if your proteins are membrane protein having that hydrophobic patches, so it may clog that transport system of that E. coli. So ultimately it will block their normal transportation, normal protein transportation, their own protein transportation. So that's why E. coli, either it will destroy your plasmid having that membrane protein cloned, membrane protein coding genes are cloned, or cell will die itself. In any way, as an experimenter, we will not get our final product. Fine.
Okay, sir. I think there's same question for codon biasness only. So she has asked that why there is higher frequency of one codon in one organism and less in other organism.
Okay, so there are different school of thought for this preferences, but it comes from evolution. Are you getting? Some means some organism preferred one sets of this codon, some organism prefers different sets of codon. Exact reason is not known, but suppose this AGA is present in some gene of E. coli, then their expression will be less. So nature have given one of the mechanism for controlling the level of protein in that cell. One group of scientists, they are thinking that this presence of different, suppose this rare codon is present in some of the gene of E. coli, then what will happen? Their expression will be less. Their protein amount will be less. Are you getting? So this presence of rare codon control the level of expression in that organism also. Some proteins having this preferable codon, then their protein amount will be more. This is one of the hypotheses of presence of different preference of this different codon.
Mm-hmm. Correct, sir. Correct. I think that is a good explanation for this, I can say. So next question is, can we use BL21C43 for production of human GPCRs?
So again, obviously that is very long protein. Scientists are trying to produce it. But see, if that protein are very long, and I showed you that if there is several hydrophobic patches, because this is membrane protein, and as you know, there are so many transmembrane region. So still we don't know the reason of not production of that hydrophobic protein in that E. coli. Sometimes by removing that hydrophobic patches we produce. Sometimes we are not able to produce, even trying all these strategies. Whatever strategies are available, whatever strengths are available, we have discussed. But so many things are still unknown. You can explore. Suppose some proteins are of 1000 kilodalton big. Then metabolic load on that cell will increase, E. coli cell. So think about their own protein, which are required for day-to-day exercise. That E. coli have to survive, na? So if your host, if you have one room and your host 10-20 host guest will come, then what will happen to that host? आपका 10-15 गेस्ट आ गया तो आप क्या करोगे? या तो आप घर छोड़ के भाग जाओगे, उस गेस्ट को ही रहने दोगे घर में, वो कर ले. Are you getting?
Yes, sir. Yes, sir.
Regions which control. Hello.
Okay. Yes, sir. Yes, sir. So I think that is a good explanation in layman terms. You can give it to anyone. So there are many couple of questions. I will ask some few important questions only.
Yes,
so,
yes. You can ask question, or sometimes you in your website, that channel also, if someone have question even after one day.
Yeah, they can easily email us and contact us. We'll surely get back to you with the answer.
Is any option on your YouTube channel?
Yes, there is. We can. Yes, but people can post their questions later on.
Yes, I will also read, or they can send me WhatsApp message also. But whatever you can ask, depending on limited time, you can ask.
Yes, exactly. So the feedback or the certificate link is already up in the chat box. So the people who are getting late can actually skip this question and answer round, and those who are interested, they can stay. We will carry on with the question answer round, and the certificate is for the e-certificate link is already up in the chat box, so you can just click over there and fill your name. E-certificate would be provided in next 7 business days. So moving forward to the question and answer round, uh,
Yes.
there is the question: we can't express membrane proteins like GPCR because bacteria don't have chaperones. So then, why don't we use Arctic strain of BL21?
No, 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 no. Still, we are using we several this membrane protein we try to express in E. coli,
Okay.
but
So can you just name some proteins which can actually membrane proteins which can actually express in bacteria
see,
like E. coli?
see, during my PhD, I have worked on Helicobacter pylori type 4
Mm.
secretion system, and
Yes.
that
Yes.
food secretion system is transmembrane channel, which is
Mhm.
composed of several proteins. So I myself have produced CAG-X protein, CAG-T protein, and several other proteins. But even we have faced the problem, but we have produced several transmembrane protein in E. coli also. Are you
Okay.
getting?
Yes. So it is possible, right? Yes.
Possible, but sometimes Helicobacter pylori is also a bacteria. So
Mhm.
pair with eukaryotic membrane protein with this.
Correct. Correct.
You getting?
Yes, sir. Yes, sir.
In
I think that's
eukaryote,
a clear explanation to this.
yes.
Then we have a question that how do CPN 10 by 60 stay active at cold temperatures?
That's see good. So this saffron scientists have taken from sacrophilic organism which grow at low temperature. अब आपको पता है कि कुछ ऑर्गेनिज्म बहुत हाई टेम्परेचर पॉलीमरेज इज पॉलीमरेज why it is working at 95 degree but normal E. coli polymerase will not work. Are you getting?
Yes,
So so sacrophilic means cold adapted organism. That gene is isolated from cold adapted bacteria which grow at low temperature. तो जो bacteria grow करता है low temperature पर, their protein will be active at low temperature only. Their three-dimensional structure in such a way that they will act as low temperature. Like that hot spring organism have that resistant protein which can act or which remain active at high temperature like Taq polymerase. Same way at low temperature, their all protein will act at low temperature. Are you getting?
yes, sir. Yes, sir. I think that's also a clear explanation for the question for the bacteria to stay at the cold temperatures. So
Yes.
I think next question is how can one know whether the promoter is strong or not?
Okay. So there are different criteria for, but simple thing is, as you know, some proteins are present in cell in high amount, some proteins are in low amount. How it is controlled. So promoter, if that promoter is a strong, RNA polymerase bind strongly, their affinities are strong, and some sequences, that conjugate sequences, similarity between the consensus sequence is same, then that is considered as strong. Are you getting?
Yes, sir. Yes, sir. Yes, sir.
In nature given different gene means different proteins are required in different amount in cell, so their promoter controls. So if that protein is required in more amount, so that gene is present under a strong promoter. So through research, scientists come to know that these promoters are strong, these promoters are weak, so they have taken that sequence and cloned into vector.
Correct, correct, correct. Yeah, I can completely understand. And even I think the question is very much well answered by the speaker. So
Yes.
next question is, what are the disadvantages of an affinity tag?
No, no, affinity tag, there is no disadvantage of affinity tag. In case of histidine tag, so histidine is, if it is present in continuous 6 histidine, then it affects the solubility of your conjugated protein.
Okay.
So अगर पहले से ही आपका प्रोटीन insoluble nature का है, उसमें और एक डाल दिया insoluble nature वाला histidine, that is 6 histidine, so it will decrease solubility, then you will not get soluble protein.
Okay.
This is limitations of histidine tag. Same like in MBP tag. So MBP tag increase solubility. This is good. It will affect solubility of recombinant protein, but in positive way. It will increase solubility. But here tag length is, affinity tag length is 44 kilodalton. So suppose if you are cloning your gene having 10 kilodalton size, तो उसका affinity tag 44 kilodalton का होगा, then what will happen to folding of that protein? That affinity tag will cover, engulf your protein. So your protein may not be active. Your protein may be covered by that affinity tag only.
Mm-hmm. Correct. So we have a similar a question which actually targets this also. So the
Yes.
question says that why polyhistidine decreases the solubility of the protein? Why does it do so?
No, continuous histidine आपको पता है charged होता है ना histidine. So due to this charging 6 continuous charged protein, वो conjugated जो से tagged protein है, your protein is tagged with that polyhistidine. Decrease solubility.
Okay, so that's the main reason.
Charging. Continuous charged amino acid.
Okay, so I think continuous charged amino acids is an appropriate answer to that question. Correct. So
imidazole ring होता है उसमें, वो भी बड़ा सा size होता है उसमें. Histidine have imidazole ring.
Yes.
उसका bulky part होता है, वो भी उसका effect करते हैं.
Correct. So, sir, next question we have is like, if we don't have much information about our protein, how can we choose the vector?
See, that's why now, if you don't have information, then you should select this pet vector. You can borrow from somewhere. You don't have to purchase. Try that gene. You should clone in that pet vector because their affinity tag is very small, so it will not hamper. But nowadays, there are so many software available so that you can check its hydrophobic character, hydrophilic character, what is the size, what is the PI, all these things. If you know the gene sequence, then you can determine all these things through in silico analysis, computer-based software.
Correct. So I think even proteomics and mass spectrometry can be answer to this question before actually choosing your own protein.
Yes, main thing is why you are choosing that cloning, that gene to clone and produce recombinant protein. That must have some information about that protein. That's why you are using that gene to clone and produce recombinant protein. Are you getting?
Yes, sir. So I think this is I think the most of the questions are really answered very well. So I think we can, like, if participants have more questions and if their questions is not answered satisfactorily,
they can note down my phone number also, and they can connect through you also.
Yes. So we will provide the email ID in the chat box.
Yes.
So they can
join on my Twitter page also, and my YouTube channel also, and they can also join your that scientific community.
Yes. So we have emailed the scientific community. I can say the email ID, and I'll just share down the social media link.
Yes, there are so many questions from admin also. Huh?
Yeah. I mean, those questions were actually from the I can say the people who actually asked in the YouTube live. So those questions were there. Yes. So I'll just provide you the link, and I think we can move forward with the vote of thanks. Up till that, I will post all the links, and the link for the certificate is already up in the chat box, so you can just fill out at that form. So do not forget to fill the form, and you can connect us on our social media handles also. That link is there. Okay. So now moving ahead on the vote of thanks.
So Dhairya, I hope I am audible.
Yes, you are.
So yes, so with the immense pleasure, I, Soumya Mishra, on behalf of scientific community, would like to end this session with the quote: "As we keep discovering our world, science blends with spirituality, and accepts we must believe in order to see. So we are thankful to Dr. Gopalji sir for joining with us and giving such a good insights for the topic for different strategies of recombinant protein production in E. coli host. We also thank our participants who have attended this webinar on YouTube, and I hope that you have been benefited from this webinar. So please do visit our website www.scientific-community.in and do subscribe for our newsletter, which you will like, which will be from which you will be benefited a lot. So please do join our community, scientific community, and help us to grow more by making such events possible. So till then, think, grow, and share. And the link for the e-certificate is in the YouTube chat box. Look forward. Thank you.
Okay. So that,
Great. So, thank you so much, Gopal sir, for coming in. And I would like to tell one thing to each and every participants that we are actually going to have a workshop with Gopal sir only on the recombinant DNA technology. So the participants who actually wants to join in can actually fill the feedback form for this, and it's already in the chat box. So I'm again typing it down in the chat box. You can just go to the feedback form, fill it, and just let us know that if you are interested in the our workshop or not. So please fill this feedback form and get the e-certificate. And the link for e-certificate is open only for next I can say 45 minutes. We will close this link down at 6:30. So please fill the feedback form link as soon as possible. And also the link to join our community groups. Also, I'm sharing it over here, so you can also join that.
okay.
Okay, so I think that's an clear idea to everyone.
Yes.
Okay, so thank you so much, Gopal sir, for such
Thank
an good
thank
insight.
you. And you keep on sending question if some questions have missed. And like
Sure,
so there are so many advantages is only purification and sometimes solubility. And this
sure, sir.
is for therapeutic proteins, drug controller will not allow affinity tag. Are you getting?
Okay.
We are
Yes,
using that
sir.
insulin, recombinant insulin protein, and that insulin have histidine tag or MBP tag. You cannot use as a therapeutic. So
Sure, sir.
you must remove that affinity tag.
Sure, sir. Sure, 100%, sir.
Yes.
Okay, thank you so much. Thank you all the
Thank,
participants
thank
to
you.
join in, and
Should I
also
leave
join
now?
us. Yes, sir.
Thank you.
Thank you so much, sir.